Hello everyone, this is Ayman Said Auda from Group 1 and today we will talk about and discuss fluid homeostasis. The animal body is composed of eukaryotic cells containing various organelles such as mitochondria, smooth and rough endoplasmic reticulum, Golgi apparatus that perform specific functions. The nucleus and organelles are all surrounded by a plasma membrane consisting of a lipid bilayer, primarily made of phospholipids with varying amounts of glycolipids, cholesterol, and proteins. The lipid bilayer is positioned with the hydrophobic fatty acid tails of phospholipids oriented toward the middle of the membrane, and the hydrophilic polar head groups oriented toward the extracellular or intracellular space. The fluidity of the membrane is maintained in large part by the amount of short chain and unsaturated fatty acids incorporated within the phospholipids. So, incorporation of cholesterol into the lipid bilayers reduces fluidity. To accommodate multiple cellular functions, the membranes are actually semi-permeable because of a variety of proteins inserted in the lipid bilayer. These proteins are in the form of ion channels, ligand receptors, adhesion molecules, and cell recognition markers. Transport across the membrane can involve passive or active mechanism and is dictated by the membrane composition, concentration gradient of the solute, and availability of transport protein. If the integrity of the membrane is disrupted by changing fluidity, protein concentrations, or thickness, transport process will be impaired. Second, in this slide, we can see examples of cellular communication. We can see autocrine, a cell targets itself, or a cell targets a cell connected by gap junctions, or paracrine, a cell targets a nearby cell, endocrine, a cell targets a distant cell through the bloodstream. Body fluid compartments. Total body water is distributed between two major compartments, intracellular and extracellular fluid compartments. And the intracellular comprises 2/3 of the total body weight, constitutes about 40% of the body weight, found in cells of the body, high in potassium, magnesium, phosphate, and inorganic anions, proteins, and low in sodium, bicarbonate ions, and chloride ions. Then extracellular fluid comprises the remaining third of total body weight, constitutes about 20% of the body weight, found outside the cells. Low in potassium and magnesium ions and high in sodium, chloride, and bicarbonate ions. Then the extracellular fluid is further subdivided into three subcompartments. Interstitial fluid, which surrounds the cells, doesn't circulate, comprises about 3/4 of the ECF, only compartment that declines with age. Second, plasma circulates as the extracellular component of blood, and it makes up about 1/4 of the ECF. Then transcellular fluid. It is contained within epithelial-lined spaces, set of fluids that are outside of the normal compartments. It includes cerebrospinal fluid, GIT fluids, bladder urine, aqueous humor, and joint fluid. Peritoneal and pericardial fluids constitute about 0.5 to 2 liters. Then we need to know about, to maintain homeostasis, we need to know about osmosis. Osmosis, membranes are selectively permeable or semi-permeable, meaning they allow some but not all molecules to pass through. Membranes of tissues vary in their permeability to specific solutes. This tissue specificity is critical to function, as seen in the variation in cell solute permeability through a renal nephron. On either side of the membrane, there are factors that oppose and facilitate movement of water and solutes out of compartments. These factors include concentration of a specific solute, overall concentration of solutes, concentrations of proteins. So osmosis occurs when osmotic pressure is present. This is equivalent to the hydrostatic pressure necessary to prevent movement of fluid through a semi-permeable membrane by osmosis. This idea can be illustrated using a U-shaped tube with different concentrations of solute on either side of an ideal semi-permeable membrane. In the plasma, the presence of proteins also produces a significant oncotic pressure, which opposes hydrostatic pressure filtration out of the compartment and is considered the effective osmotic pressure of the capillary. And Stirling forces. The oncotic and hydrostatic pressure are key components of the Stirling forces. Stirling forces are the pressures that control fluid movement across the capillary wall. Net movement of water out of the capillaries is filtration, and net movement into the capillaries is absorption. There are four forces controlling fluid movement: the capillary hydrostatic pressure, and the capillary oncotic pressure, the interstitial hydrostatic pressure, and the interstitial oncotic pressure. Then we turn to homeostasis. To maintain fluid balance, fluid input needs to match fluid output. If intake from food and beverages exceeds output, which from urine, fluid in feces, and sensible losses, the organism is in positive balance, and urine volume will increase to eliminate excess fluid. Negative fluid balance occurs when intake is less than output. In this case, integrated responses will increase thirst and decrease additional fluid losses until homeostasis is restored. On the cellular level, homeostasis is possible due to expandable semi-permeable membranes, which can accommodate small changes in osmolarity via osmosis. However, for proper cellular function, the intracellular fluid and thus osmolarity must be kept under tight control. Measurement of fluid compartment size. The indicator dilution method is used to determine the volume of fluid in the different fluid compartments. This is done using indicators specific to each compartment. A known quantity of the substance is infused into the bloodstream of the subject and allowed to disperse. A plasma sample is then obtained and the amount of indicator determined. The compartment volume is then calculated by this formula. Volume, in liters, equals amount of indicator injected by milligrams divided on final concentration of indicator. Then the compartment can be indicated by total body weight, can be indicated by antipyrine or tritated water. The extracellular fluid can be indicated by inulin. The plasma volume can be indicated by Evans blue dye, which binds to plasma proteins. The total blood volume is composed of the plasma and RBCs, and the hematocrit is the percentage of RBCs in the whole blood. So, this is our references, and thank you. This is Ayman Sayed. Hope it was useful for you. See you at the live session.